生物书曾告诉我们，视网膜只是眼睛的感光底片，而他的研究发现，视网膜还具备信息处理的功能。他不仅学术成绩斐然，还将自己设计的实验设备开源，让更多条件受限的研究者获益，以实际行动支持科研的透明化和开放化。他也是首届腾讯与自然科研共同发起的自然科研全球影响力大奖的获得者。
Let's welcome Professor Thomas Baden.
Good afternoon. So I'm a neuroscientist, and what interests me in particular is how the nerve cells in the brains of animals and the connections between them compute, and ultimately how they can give rise to behavior. Now, in order to do that, what we need to do is we need to be able to measure the activity of many neurons at the same time in the live animal, and we need to be able to relate this to a particular task that the animal is trying to perform. That is not an easy thing to do. And one of the tools that we have in modern neuroscience available to us is the use of genetically engineered model systems. And you see one of those guys behind me. So this is a baby zebrafish. And what's so special about the zebrafish is that it is almost transparent at this age. Now, this is incredibly powerful because it basically means that without having to do very much at all to the animal, we can directly see the brain. Now, of course, if you go out into the wild and you catch a zebrafish, the brain isn't green. The reason that this guy's brain here is green is because it's been genetically engineered to be that way. And basically what happens is that the nerve cells inside the brain have been programmed to express all of the things that they usually express, plus one more thing, which is an extra protein, which happens to fluoresce, in this case, in green. But it actually goes one step further than this. This particular protein doesn't just fluoresce in green to reveal the structure of the brain, which does so very beautifully in this case, but it actually changes its fluorescence properties depending on the activity state of the neuron that it is in. So if the nerve cell is active, effectively you get a brighter fluorescent signal from specifically that cell, and if the neuron stops being active, that signal goes back down. So you can imagine that having this kind of tool available is very powerful. Now, for me personally, I don't just work in the brain in general, but I work on our sense of sight. So for this, we need to look into the eye of the animal. Now, if you do this, you see the schematic here behind me. This is what it looks like when you slice a zebrafish eye open at the midpoint. The big green blob, that's the lens, that's the optics of the eye. And pretty much everything else that you see inside the eye cup is nerve cells. It's basically a little brain inside the eye. So what we can do with this kind of setup is we can now express these activity marker proteins in specific cells within the eye, and then we can shine light into the eye of the animal while it is alive, while it's processing the information, and we can watch the nerve cells in the eye do their job. And this is what this might look like. The stimulus in this case is just a flash of light. We switch on the light, we switch off the light, and we repeat. And what you can see is that the different layers of nerve cells in the eye process this information and respond not all in the same way, but in different ways. Some respond when you switch on the light, some respond when you switch off the light, and some do something completely else. Now, we can do these kind of experiments in the eye. We can also do them in the brain. And this is what it would look like if we did the similar kind of experiment with a very expensive microscope, where we take 3 different slices, optical sections, through the live brain of the fish. We present the same stimulus, and again we see some neurons switching on, switching off in relation to the light, plus we see a huge number of neurons doing something completely else. This should not surprise us. This fish is alive, it's presumably thinking, it's presumably planning. So if we have these kind of tools, and we can now watch the activity of literally thousands of neurons at a time, and we can manipulate the environment around the fish, we can actually manipulate the activity within these neurons, this is going to be a really powerful technique. It already is a powerful technique, but I think it's going to become even more powerful over the years to come to understanding how brains work, first in little animals like this, and then going outwards until eventually we maybe understand something about our own brains. Now, this pursuit of scientific knowledge, I think, is very exciting. But here comes the plot twist. To be able to do these kind of experiments, you need a huge pile of money. And the result of that is that very few scientists around the world get a chance of doing them. And that's a big problem. If you really want to make headway in problems such as how the brain works, we need to have as many scientists as we can thinking about these kind of problems, and therefore we need to give those scientists the tools. So to give you a scale of this problem, this is what the world population looks like mapped out over the countries. Some countries are more heavily populated than others, but overall, the entire world is full of people. If we now do the exact same thing for the number of scientific papers, and this is now in any discipline, that come out of these countries, the picture is radically different. The vast majority of papers come from the global north. The global south contributes substantially less. For example, if you focus your eyes on Africa, the continent has almost disappeared. This gets even more dramatic if we look at Nobel Prizes. So clearly, this is using human capital around the globe in a terrible way. We're giving a very small number of people the tools to do the biggest research. What can we do to equalize this? Well, one very obvious route is you can throw lots of money at the problem. And presumably that would go partway into addressing some of these issues. But in the famous words of Rutherford, we haven't got that money, so we'll have to think. So what can we do? So these kinds of experiments that I was showing you as an example, the reason they are so expensive is because you need to do 2 major things. One, you need to buy and calibrate a very expensive microscope. That's the thing on the left, 2-photon microscope. This will cost you something like $200,000. And then even once you have that microscope, you still need a sample to look at. So you need the ability to genetically manipulate model organisms. Now let's look at that second one first, genetic manipulation. If you go into a standard molecular biology lab, these are the sort of machines that you might find standing around. Each individual machine of those will cost a minimum of $1,000, many of them substantially more. So what do? Well, the first one is basically an oven. It's an incubator. The main job of this machine is to keep temperature and humidity at a certain level, and that is critical to keep whatever biological samples you have alive. Next guy, that's a centrifuge. Absolute key technique in, well, many sciences, allows you to separate samples by basically accelerating gravity, by spinning it. Here we've got a PCR machine that's basically a genetic material replication machine. The way it works is you've got a bunch of different enzymes that work at different temperatures, and you go through cycles of going to different temperatures that are optimal for these different enzymes, and that will generate, well, will facilitate the replication of genetic material. So basically, it's a glorified heating and cooling device. The last thing here, that's an electrophoresis chamber. Again, this we use to separate samples, but now in a different context. And now we do it with electricity. You basically apply a current. You've charged particles. Depending on the size and the charge of a particle, they will move at different speeds, and therefore you can separate them. So these kind of tools are absolutely everywhere when you go into a well-funded molecular biology lab. But if you think about the physics behind what these machines achieve, the physics has been solved for more than a century. This is not new stuff. This is the sort of stuff that surrounds our everyday lives we don't even think about anymore. So that being the case, can we maybe go to our garage and knock up some of these tools from scratch just based on what we see flying around? I think we can. So this is an incubator, styrofoam box and a light bulb. It will give you a reasonably stable temperature. You can hatch your eggs. This will work. If you want to build a centrifuge, there's so many things out there that spin. You can use a bicycle wheel. You can use a fidget spinner. Many tools spin. Many tools spin fast enough to separate samples in a meaningful way. Similarly, if you need controlled heating and cooling, why not take a bunch of water baths and just take your sample and dip it iteratively through them? This is actually how this device was invented in the first place. If you want to zap, you could just take a lunchbox and batteries. It works just the same. Okay? So you can imagine that these kind of tools are much cheaper. Now, have we solved it? No, unfortunately not, because the problem with these tools is they're not particularly reproducible. Right? If I do my experiments with any one of those tools 20 times, chances are I'm going to get some different results over time. If I write this up in a paper and then you try to reproduce it, then presumably you're going to get different results, because it's really hard to get those machines to be always the same, and that's critical in science. In comes the 3D printer. So this really has been the game changer. Because with a 3D printer, what I can do is I can design a very precise mechanical part, which I need for my scientific machine that I want to build. I can test it, and then once I'm satisfied that this is good, I can take this file, I can put it online, then you can download it, stick it in your 3D printer, and you get exactly the same part. That means that reproducing the mechanical aspects of these machines has become massively facilitated. And I should point out, to print anything on a 3D printer like this, it will cost you a fraction of a dollar. The material that you need for these printers is incredibly cheap. So here's an example of a 3D printer at work, linked to a publication that we had a few years back, where we drew attention to the possibilities of using 3D printers in this way. And one thing that we did, just to make the point how this is possible, is we showcased a 3D printed pipette. Now, pipette is an absolute corner piece in any molecular biology lab. It allows you to transfer samples of whatever material you need to be transferring. So we put this 3D printed pipette in there. It was a reasonably good pipette, but it wasn't yet up to the standard that you might find in a commercial one. But critically, we put the plans of this pipette up. And I should point out that we didn't invent this pipette. I saw a different pipette online. We modified it, and we put this one forward. So then we put ours forward, and then others saw this pipette, and they say, hey, this is great, but you did this part really stupidly. You should change this bit. And the beautiful thing about that is they didn't need to tell us that. They could just take the design, change that bit, publish their own pipette, say, look, these guys' pipette is this good, mine is better. And then the next guy comes along and says, okay, this pipette is great, but I can make it better still. So today, when you look around, when you go in a search engine and you put 3D printed pipette, you will find lots of different versions, lots of different quality grades. Here's a few of them displayed. We come to a point when it's not even that easy anymore to tell apart which one is the commercial one. It's the second from the right in this case. And actually, some of these pipettes are now meeting ISO standards for accuracy. So this is critical. So this is the standard that you would need if you wanted to commercialize this pipette. You could sell this, and it would fly, right? Similarly, you can say, I did my experiment using these pipettes, and then people wouldn't really have grounds to complain that you used subpar equipment, right? So some of these tools are really coming along, and they're becoming very powerful. This is another context that may have come across in terms of 3D printing. This is people designing mechanical parts that can replace limbs. So for example, if someone loses a hand, you can 3D print this thing, and the fingers, in this case, are connected with strings. If you pull on the string, the finger contracts. So it's a sort of primitive prosthesis. You can imagine if you combine that with a motor and maybe a muscle electrode, you can get the person to actually control the movement in this hand. And again, very cheap to build. So this is sort of where we can push the 3D printing at the moment with these kind of filaments. The problem, however, is you can print as much of a brain as you want. It's never going to start thinking for you. So really, instead, what we need is we need to combine these 3D printing possibilities with consumer-oriented electronics. Things like this guy. This is a microcontroller. It's basically a device that allows you to connect your computer to a piece of electronics. In a simple way, you could say maybe I press a button on my computer and then a light switches on. Or I will have a sensor connected to this thing. The sensor senses something and sends the signal to a different electronic piece to maybe open a door or something. So with these kind of little mini brains, if you combine them with the 3D printed, now we're talking business. So here is one of the possibly silliest things you can do with this. You may have come across this. It's a so-called useless box. You switch it on, and the sole purpose in life of this box is to switch itself off again. So you can do that, or you can do something useful. So we've tried to stay on the useful side. So here is a proof of principle that we've tried to come up with, combining 3D printing and cheap electronics. And this was very much inspired by the $100 laptop idea that you may have come across some years ago. This is the $100 lab. So this is basically a 3D printed platform for microscopy. It's a fluorescence microscope. It also has some additional capabilities, which I'll show you in a moment. And really, this was a proof of principle that for 100 euros, you could build a machine that could really meaningfully contribute to the research going on in a modern lab, even a lab that's well-funded already. So just some examples of what you can do with this. So here you see our friend the zebrafish again, 2 examples. Here's the video. So on the right, what we can see there is this is the zebrafish, transparent as I pointed out, and the thing that's moving, that's the heart. So this heart is expressing a fluorescent protein, and it lets us visualize the contractions of the 2 chambers. It's a fish, so it's got 2, not 4. And you can imagine this becomes immediately useful if you, for example, want to study circulatory function. Oops. OK, the other video didn't play. So we're absolutely not alone in generating these tools. This is a global effort. It's a huge community, and it's a very rapidly growing community. And this community meets at conferences, they meet online, and they discuss ideas. They discuss what are the standards that we need to set, what are the projects that need addressing. And this has started to get the attention of a lot of media over the last years. So here's one coverage that we had recently from one of those conferences, basically reporting on the idea that people are coming together to build low-cost, open hardware scientific tools. Now, all this time I was talking about this molecular biology aspect, but what about the other half, the 2-photon microscope? Well, the same principle applies. Even though this is a very complicated machine, these sort of machines are fundamentally modular. That means that I can take one module, where maybe I have an idea of how to improve it, how to make it cheaper. I improve it, I publish this, I put it into my microscope, and I've saved maybe 5% of the costs. And then someone else comes along and thinks, oh, maybe this module, I can change it. I take it, I modify it, put it back, and we've saved another 5%. So if we keep doing this, we can really push down the cost of these kind of microscopes. So already now, if you go for an open hardware approach to building one of those, we've slashed the cost by more than 50%, and this trajectory is going down rapidly. Also, you should take these things with a bit of a pinch of salt, because two-photon microscopes don't look like the thing I showed. This is what it actually looks like in our lab. And I just want to highlight a few things. We improvise a lot. So ours are basically held together by hope and tape. So we've got tape, we've got LED strips, we've got putty, we've got a soda can. Turns out that's really good for electrical shielding. So we're combining the low-cost and the high-cost approaches in the lab. And this is actually the microscope that generated the picture I showed you in the beginning. So maybe instead of using money, we can use an open hardware approach. And just in closing, I want to highlight that it's nice to stick these things online, but you need to get people to actively search out these possibilities and to learn about these possibilities. And to do this in particular with a focus in Africa, colleagues and I founded this organization, Trend in Africa. We've got projects in more than 20 countries by now. There are lots of different types of projects, ranging from community engagement. We've got volunteering programs. And one of those things is promoting open hardware. And what we do there concretely is we go to our African partner universities, and we get students, and we show them, look, this is a 3D printer. This is a microcontroller. This is where you can find information online. We do a crash course in these kind of things, and we encourage them to build the scientific tools that they need for their own research. So in this case, for example, here they built a behavioral chamber for rodents. This thing is published. You can look it up, and you can build it yourself in your lab. So now we've come around full circle, where now the global south can generate the scientific tools that maybe the rest of the world needs. So in closing, I want to change a little bit this Mandela quote. It's very famous. Education is the most powerful weapon which you can use to change the world. Well, I think it needs a twist. I think it's education to help yourself. Thank you.